Hey guys, so here we are going to start with the topic of paper chromatography. We are now going to solve one of the past paper question related to paper chromatography. I am going to write down the answers. Before writing the answer, listen the question very carefully and read it carefully. Leaves of plants contain a number of different colored pigments. Four students want to extract and analyze some of these colored pigments. Okay, the leaves are chopped up and ground using a mortar and pestle and then mixed with ethanol. Okay, so right now I would like to inform you that examiner can ask this thing in a paper that which is one is pestle and which one of them is mortar. Okay, so be very careful about this as well. The pigments are separated using paper chromatography. Okay, done. Suggest a reason for cutting and grinding the leaves. Okay, for chopping the leaves, what is the reason behind that? So that the pigments can be taken out of the leaves very properly, so that the pigments from the leaves can be taken. Okay, so any answer related to that, but be very aware of this thing that we our target is to take the pigments out of the leaves. We know this thing that it is mixed in ethanol. Okay, why ethanol, why not water? Because we know this thing that plant material, that leaf material is insoluble in rain. Water, we have observed this thing that whenever there is raining, no green color is falling on the ground. It means that plant material, that leaf material is on insoluble in the rain. That's why it is not dissolving. So what we have to do, we have to dissolve that plant material, that leaf material into a solvent, and the solvent selected is basically ethanol, that and alcohol. Okay, the pigments are separated using paper chromatography. Suggest a reason for cutting and grinding the leaves, that I have told you, so that the pigments can be taken out of the leaves. Now there are four setups. You can see one of the setup is A and other setup is B here. There is a third setup called C, whereas the fourth setup is D. Okay, let's look into these all four. Okay, four students do the paper chromatography separation. Okay, A part 2. The diagrams show four sets of apparatus used by the students. Three of the students make mistakes in setting up the apparatus. Okay, let's see what are the mistakes. Okay, I would like to tell you that examiner is asking about these mistakes on the next page, but right now I would like to tell you what is the problem and where is the problem. Okay, so in A, let's look into the A carefully. Okay, we can say this thing that in A there is a baseline which is not only drawn in ink. So this is the problem number one. It is drawn in ink. Rather, it is immersed in the solvent as well. Okay, it should be above the solvent, but it is in the solvent. We can say this, so this is another mistake. Okay, so there are two mistakes in A. Okay, let's look into the B. B does not have any lid on it, so this is the problem number one which we can see. And number two is this, that the spots of B are also drawn in ink, so this is the problem number two. Let's look into C. Okay, this is the pencil, the drawing is with a pencil. Perfect, we are very clear on this, so it is correct. Okay, but lid is missing, so this is a problem. This is the problem number one. And there is another problem, that is the baseline is drawn in the solvent. The baseline should be above the solvent, but it is drawn in the solvent, so this is the problem here. Okay, so I am marking a cross here as well. Okay, now we can see that we can see the D point. Okay, the lid is there, correct. The line is drawn with the pencil, correct. And the third is that the line should be above the baseline, that is correct too. So it means that we can see that these are the mistakes in A, B and C, and there is no mistake in D point. Okay, let's move on to the next page. Okay, so it is, I am cancelling this and discard the changes. Okay, so I am on the next page. Okay, so right now I am going to edit this as well, and this is okay. So which diagrams A, B, C or D show the correct setup for the apparatus? So it is the answer D. We will write D over there. Okay, and then describe two mistakes. They are just asking two mistakes. Rather, we have found three mistakes in the A, B and C. One of the mistake was this, this, that lid was not present, and we have to mention this over here as well, that mistake number 1. Okay, so we have to mention correctly the look at the question number 3 again. Describe 2 mistakes made by the students shown in the diagrams. Explain why each mistake will prevent the chromatography from working correctly. Okay, so mistake number 1, you can say very clearly that in setup B, comma, lid is missing. I am writing it here very properly here. In setup B, comma, lid is missing. Okay, here I am writing alcohol, or you may write ethanol very clearly, the one which the word which they have used actually. Ethanol solvent will evaporate, hence experiment will be affected. Experiment will not give correct result. Okay, mistake number 2, we can say in C, or you may say in A. Okay, in A, let's say in A. In A, the baseline slash the start line is drawn in ink. Being soluble, this is the explanation. Being soluble in the solvent, ink will not give the correct results. So this is the answer for these 2 questions. Okay, chromatography often uses water in the beaker. Suggest why ethanol is used instead of water in this experiment. So what is the answer? As plant pigment, as leaf pigment is insoluble in water, as leaf pigment is soluble in ethanol, that's why ethanol is selected for this. Okay, now I am on point 5. What property of ethanol makes it hazardous to use in laboratory? What safety precaution need to be taken to avoid this hazard? Okay, so what is the property? Ethanol is flammable. Okay, this is the property. And what is the precaution? Ethanol should be kept away from flame. Okay, away from flame. This is the answer here. Okay, now I am turning the paper to the next one. Okay, and this is it. Okay guys, you can see this thing, that this question is of 14 marks. Okay, so one should pay a perfect attention to this question. Now the diagram show the results for 3 known pigments. Okay, and W and then X and then Y. We can see this thing, that all 3 are shifted into single spots. It means that W, X and Y are considered as single spots, or you may consider pure substances. But cabbage is shifting itself into single spot. Okay, whereas grass and spinach are converting themselves into more than one spot. So grass and spinach will be considered as mixtures here. Okay, this is a point one should pay attention on this. Okay, let's coincide the spots with each other. Okay, I am trying to change the color. Yes, so here X is matching with cabbage, and X is matching with grass, and X is matching with spinach as well. Okay, so we can say that X is present in the cabbage, X is present in the grass, X is present in the spinach. Okay, let's find out about W. W is matching with grass and W is matching with spinach as well. It means that W is not present in cabbage, whereas W is present in grass and spinach. So only one substance is present in cabbage, and what is that pigment? It is X. Okay, let's discuss about Y now. Y is present in only one, and that is called grass. Okay, so grass and the spinach are the two which are containing more than 2 substances. Okay, let's discuss further. Okay, how many pigments are there in grass? So we will say three pigments are there in grass. How we know this thing that there are three pigments in grass? They are saying explain your answer. Three lines are for this question, whereas mark is one. So what will be the answer? Three pigments are there in the grass, as grass is shifted into three different spots. Okay, which pigment in is in all of the plants? Okay, which is in all of the plants? We can say this thing that X is present in cabbage, grass and spinach. So we can say X is present in all three. Okay, calculate the RF value of W. Okay, let's calculate the RF value of W. Here I'm changing the color to green. Okay, let's mark the line in the center. This is the distance traveled by W, whereas this is the distance traveled by solvent. Okay, so we will calculate the RF value of W. Distance traveled by W divided by distance traveled by solvent. Which of the plant contains pigment W? So W is present which of the plants? Look, they are writing plants. Okay, so you have to mention both of the plants. Sometimes the students are writing grass is containing W. No doubt grass is containing W, but examiner is asking which of the plants. So you should say grass and spinach both contains W. Okay, so this question is telling you that sometimes a very simple topic can be asked in a very long way. It's a 14-mark question, so a student should pay a perfect attention on the topic of paper chromatography as well. Thank you very much.